All right. Well, thank you all for being here. I will present my our research in Parkinson's disease using fNIRS, and the first paper I will discuss is this paper in which was published in Neurophotonics. Um, and the motivation for this study is because people with Parkinson's disease they experience changes in fine motor skills, and that's one of the hallmarks of the disease. fNIRS is very apt to study this because it's non-invasive, it's portable, so it's a promising tool for assessing those changes in the brain that are related to fine motor skills. Our population who participated in the study was 20 patients with the disease and 20 matched controls. Just as a brief introduction, so Parkinson's disease is a type of movement disorder. It's characterized by a slowness of movement, stiffness, tremor, and it's caused because of the loss of dopaminergic neurons in the substantia nigra. Of course, all the studies were approved by the local ethics committee, and the probe that we used was this setup. It was planned to cover the precentral gyrus, the postcentral gyrus, which span the motor cortex M1. And to study the brain function during a fine motor task, we designed an experiment which involved two types of finger tapping with the left and the right hand, and they were spaced with a variable interstimulus period, which was to prevent that the task became phase locked with the physiological hemodynamic oscillations. This is the general overview of the data processing. I won't go into details. I just want to mention that we made sure that the quality of the signals in every channel was good enough. We also performed detection and correction of motion artifacts, and we did pretty much the standard pipeline to study a task-based paradigm. I'm going to focus on this part. Later we come back to the resting state part, and we use a GLM to estimate the hemodynamic response function. These are the clinical data from our population. We can see that the controls are very well matched. There is a significant difference in cognition. As expected, our group with Parkinson's disease presented a slight cognitive impairment, which is also one of the hallmarks of the disease. First, we determine which channels were active in each of the two tasks, and we can see that especially in controls, the activation zone is larger for both tasks. In the group with Parkinson's disease, we saw only one channel with significant activation. So all the group comparisons were performed between this channel and the corresponding data in the control group. I have to say that all the participants reported right hand as their dominant hand. No one said that they were left-handed. This is probably due to the fact that they are senior citizens, and back in in their time, teachers in the school forced left-handed people to write with their right hand. So we consider all of them right-handed, and when they perform the finger tapping task, we see a delay, hypoactivation in the group with Parkinson's disease as compared to the controls. The peak in their response shows up a little bit later, while we examine the left hand finger tapping task. We saw also delayed activation, but this time it was exacerbated in the Parkinson's disease group. They presented a larger activation. This is well, we interpreted this as a compensatory mechanism for a more difficult task. Then we try to see some associations between the clinical data and the fNIRS metrics. We saw that the higher their cognitive level was, the faster their hemodynamic response was, both in oxygenated hemoglobin and total hemoglobin. We also saw that the progression of the disease, as assessed by the Hunjar scale, also the higher the scale, that is the more severe the symptoms were, the larger the hemodynamic response in HBR. We also see that this is the levodopa equivalent daily dose. That's the medication that they usually take for treatment. And we see that the higher the dose, also the more delayed the activation. Finally, we also begin to investigate the functional connectivity. Functional connectivity, that is the synchronicity between different parts of the brain that are functionally related, although they may be anatomically apart. We can see visually that the control group has a stronger functional connectivity, denoted by these warm colors, as compared to the Parkinson's disease. And we see that several channels show a significant difference in connectivity. We also measure the connectivity between the same hemisphere, the intrahemispheric connectivity, and across hemispheres. We found that there is a significant decrease between the connectivity between left and right hemispheres in the Parkinson's disease group, as compared to the controls. So this interhemispheric connectivity, we think that's it's crucial for a good motor function, and all the motor impairments in Parkinson's disease are reflected as disruptions in this connectivity. We also found that the delayed activation, that might be the explanation of why it's more difficult for patients with the disease to perform this fine motor task. In our second study, we continue to explore functional connectivity in Parkinson's. We use the same data, and we wanted to investigate how the disease affects brain networks, in particular the motor cortex. And we basically found this. Also a decreased functional connectivity overall, but we investigated some more parameters. Now we will use the same data, all processed with this arm of the resting state. We bandpass filter the data in the functional connectivity band that we know that this connectivity is present in these low frequency oscillations. We perform here I have a mistake. It's not the the global signal that we regress, but the signal from the average of the short channels, and that was done to minimize the influence of superficial hemodynamics in our connectivity data. And the measure of connectivity that we performed was Pearson's correlation, which was then C-transformed to perform the statistical tests. So we took this weighted connectivity matrix, and we also wanted to investigate some more metrics, all these metrics based in graph theory. So we assessed three metrics. The first was the clustering coefficient that gives you an indication of interconnectivity in the network. We also computed the small world index. That's a simple measure of the combination of non-random clustering with near random path length. Finally, we also computed the characteristic path length. That's the average number of nodes that must be traversed to connect any one node in the graph to all the rest of the nodes. And as a second metric, we also perform network-based statistics. That's more or less, it's not exactly the same, but it's more or less the equivalent in graphs of the statistical parametric mapping cluster that we see in the fMRI images, that we have a significant difference in activation in one cluster of boxes that are interconnected. Now we see the significant differences in connectivity between two populations, but reflected in a graph component. That's the components of the connectivity matrix that are linked together. So here we see this not connection by connection, but as components that differ significantly between the controls and the participants with Parkinson's disease. So the first result, that's a statistical comparison of connections between the two connectivity matrices of each group. That's done with a standard method. Perform a statistical test between each connection, and we correct for false positives using false discovery rate. So we ended up with these different connections that the most different was found in the right hemisphere, here between the 5 and the channel 5 and channel 7, channel 5 and channel 9, which cover the post-central and the pre-central gyrus. They were significantly reduced in the patients. That was the most different connections that we found here using the standard method of comparing one by one and doing false positive correction. Then we took this weighted matrix and we converted this weighted matrix to a matrix of zeros and ones with different sparsity thresholds. And at each threshold, we did the comparison here for the clustering coefficient. We see that there are significant differences at different thresholds, at many different thresholds. And if we compare the area of under the curve for this metrics across several thresholds, well, we see a significantly lower clustering coefficient, which suggests that in people with Parkinson's disease, there are networks that are less tightly connected in specific regions. Then our second metric, which is the small world index, we also saw that there are significant differences across different thresholds. And if we consider all these metrics across all these sparsity thresholds, we found also a reduced small world index that indicates, well, a deviation from the optimal balance of specialized and integrated information processing, which also reflects decreased network efficiency in people with the disease. Finally, this is the significant group of connections that differed significantly in the Parkinson's disease group. And that's one network with 10 nodes and 9 edges. And we saw that the greatest number of these connections originate in the left postcentral gyrus, here in the channel 22, and it's mostly bilateral, although there are a number of connections that appear in the left hemisphere. So this disrupted network that includes areas involved in both motor control and sensory processing, which may explain the deficits that appear in the disease. So for this work, we found that both the clustering coefficient and the small world index were significantly reduced in people with Parkinson's disease. We wish that we could state that this is a neuroimaging biomarker of the disease, but it's not the case, because this kind of decreased connectivity metrics are also found in other diseases, for example, in Alzheimer's. But we can use these network biomarkers to, not for diagnosis, but for monitoring the progress of the disease or the efficacy of the treatment. The main limitation of this work was that all the thresholds were based on the literature, so this procedure was subjective. We didn't have an objective method of choosing like the optimal threshold. And finally, in our third paper, now we progress from assessing hemodynamics, studying functional connectivity, and now we are using this all these rich data to integrate fNIRS signals in a machine learning model that can distinguish patients with the disease from controls. So we took all the data from resting state data, and we extracted features. So these features represent the resting state data, and we have two types of features. We have features in the time domain, which are these 6 features that we computed for each channel and for each type of hemoglobin. We also looked at the signal in the frequency domain, and we also computed 6 features for each channel and for each type of hemoglobin. So we have a total of like almost 800 features. As a pre-processing step, we standardize the features. That is, we made all the features with 0 mean and unit variance. And we removed also some highly correlated features that are well, basically redundant. And we performed 2 sets of experiments. So in the first set of experiment, we took all these features, both the temporal and the frequency features, and they were ranked by these algorithms, and then we selected the top 1% of these features to train a classifier. We chose in this paper, we chose a simple classifier. It's always a logistic regression classifier, and we measure the accuracy of this classifier based on the ranking of these features. So we repeated the process for all those features, and they were ranked at the top 1%, then the top 2%, and up to the top 10%. So this resulted in 10 subsets, and then we chose, we used all these 10 subsets, and we chose the subset with the highest accuracy as the optimal subset. Then in the second experiment, we didn't do the ranking of the features. We took the original features, and we created various subsets through 2 different algorithms. These subsets that we obtained were used to train a classifier, and then the classifier with the best performance was selected for the optimal feature subset. Then, now that we have the optimal subset, we perform the classification to give the final metrics of our experiments. So how we assess the performance? We use the k-fold cross-validation. So what we did was we partitioned our set of feature vectors into 10 folds, and we used 9 folds to train the classifier, and the remaining fold was used to measure its performance. And we repeated the process 10 times to evaluate the classifier with each unique fold. Finally, all the performance metrics were obtained by the average of the performance at each fold. And these are the results. We used, as a performance metric, we used the accuracy. That's the percentage of hits, of correct predictions. We also use precision, also called positive predictive value. That is the probability of having a true positive when the classifier gives you a positive result. We also used sensitivity. We called it recall. That's the fraction of relevant instances that we found. We performed also the geometric mean of precision and recall, the F1 measure, and we use the area under the curve of the receiver operating characteristic curve that combines specificity and sensitivity. So with the first experiment, using feature ranking plus the the wrapper feature selection subset and the genetic algorithm, we obtain very good measures with a reduced subset as compared with experiment 2. In second experiment, we didn't rank the features and we obtained not very optimal results. Also, a more complex model because the subsets of features were larger. So the best method is this, which can accurately distinguish between people with Parkinson's disease from healthy controls. So in this paper, we found that there is a distinct pattern of hemodynamic activity in patients with Parkinson's. We can distinguish them from healthy controls with high accuracy, depending on the model, and the most accurate detection was obtained with logistic regression classifier using either 14 or 16 features, depending on the number of faults used in cross validation. So we show that we can use fNIRS data and machine learning models to detect Parkinson's disease in a non-invasive way. Of course, all the works that we do here, we like them to be open, so you can check them the data at this address. Also, the source code for the papers, and everything is open for you to check and hopefully find new insights using our data. So these studies in Parkinson's, well, we think that this delayed in hemodynamic response function that might serve as a biomarker of the disease, perhaps for diagnosis, but also for following the progression of the disease. We found that functional connectivity is in general decreased in Parkinson's using different metrics: interhemispheric connectivity, small world index, etc. But unfortunately, this is not a characteristic exclusive to Parkinson's disease. It also happens in some other neurodegenerative diseases. We can use machine learning and integrate it with fNIRS data to discriminate, to distinguish patients with Parkinson's with high accuracy. But of course, we have limitations in this study, like the limited coverage of the fNIRS probe, the optoid separation, also the sample size is limited. This is not a very large sample; it's more of a pilot study. But we think that this type of studies have the potential to become longitudinal. We want to expand fNIRS to other neurological disorders and continue the integration of machine learning methods with fNIRS data. Well, thank you all for being here, and thank you for the invite.